This is a video about the A-level chemistry topic of isomerism, which appears in the Introduction to Organic Chemistry topic and is examined in paper 2. Isomers are molecules or polyatomic ions that have the same molecular formula. In other words, they have the same atoms of each element, but they have different arrangements of those atoms. So based on that definition, which of these molecules are isomers of each other? This might be easier to answer if you first write down the molecular formula of each molecule. It's easy to discount the last molecule because it's the only one that contains a bromine atom, but we also need to think about the number of each atom. The correct answer is that 1 and 3 are isomers. Even though 2 is a hydrocarbon, it has 2 additional hydrogen atoms, so it's not an isomer of the other 2. Additionally, isomers have different impacts on the body. The classic example is thalidomide, which has 2 enantiomers or mirror image isomers. The R enantiomer is a sedative that was also found to reduce the impact of morning sickness, so it was actively prescribed to tens of thousands of expectant mothers. But the S enantiomer is embryotoxic, and this was also present in the medicine, and this led to the birth of over 10,000 babies with severe deformities. There are also less deadly examples. Carvone also forms 2 enantiomers. R-carvone tastes and smells like spearmint, while its mirror image, S-carvone, has the taste of caraway seeds. We can break isomers down into different groups, starting with structural isomers. These include chain isomers, functional group isomers, and positional isomers. Structural isomers are molecules that have the same molecular formula but a different structure. For instance, these 2 molecules are both C4H10, but one is butane where the other is methylpropane. They are chain isomers because their longest carbon chain is different lengths, although their functional group is the same and they're both alkanes. Next, we have the functional group isomers. Remember, the functional group is the atom or group of atoms and bonds that give a particular group of molecules their chemistry. As we mentioned, alcohols can be oxidized to make aldehydes if they're primary alcohols, or ketones if they're secondary alcohols. This here are propanal and propanone. They have the same molecular formula, but the functional group is different. Another example would be hexene and cyclohexane. They're both C6H12, but due to the cyclical nature of the second molecule, it doesn't need a double bond, and this affects the chemistry. The molecule on the left would decolorize bromine water, but the molecule on the right would not. Then we have positional isomers, where the functional group is the same, just in a different place. So the longer the main carbon chain, the more positional isomers you can have. These are propan-1-ol and propan-2-ol, the molecules that are oxidized to make propanal and propanone. As you can see, they both have a hydroxy group, but they're in different positions. Similarly, if we think about alkenes, we can move the position of the double bond to make pent-1-ene and pent-2-ene. A typical exam question might ask you how many structural isomers there can be of a particular molecule. So here we're thinking about C3H6Fi. The first thing that I'm going to notice is that this molecule cannot have any chain isomers. And the reason for that is that it only has 3 carbons in it. So if you think about a normal propane chain, if we were going to have a chain isomer, it would have to be a C2 based molecule, and then that extra carbon would need to be a methyl group coming off it. But because it has a main chain that's only 2 carbons long, as soon as you add that methyl group, you're back to a chain of 3. The next thing that we're going to notice is that this is a halogenoalkane or a haloalkane, and the functional group here are the fluorine atom and the iodine atom. So we can't have any functional group isomers because they don't have a different name or a different functional group if they're found in a different place in the way that, say, an aldehyde or a ketone group would. So now we can start thinking about how many different isomers we actually have. We know that the basis of our molecule is going to be this propane 3-carbon chain, and then it's going to be some manner of fluoroiodopropane. So what we need to decide next is where are those atoms going to be. So we could put our fluorine atom on the first carbon, in which case this would be 1-fluoro something iodopropane. or we could put it on the second carbon, but we can't put it on the third carbon, or rather we can, but if we did that, then we would actually just start numbering from the right instead of the left, because we're always numbering things in such a way that we're using the smallest possible numbers. There isn't a convention where we always work left to right or anything like that. So right now I've got two options for where my fluorine can be. Then I can think about adding in my iodine. So I could also put my iodine on that first carbon because the carbon can make 4 bonds, so it still has plenty of room. So in this case, we could have 1 fluoro, 1 iodo propane, or we could put that iodine on the second carbon, in which case we could have 1 fluoro, 2 iodo, or 2 fluoro, 2 iodo, that would be fine as well. And we could put it on the third carbon, but only if our fluorine is on the first carbon. So we can have 1 fluoro, 3 iodo, but we can't have 2 fluoro, 3 iodo, because in that case we would again start counting from the right hand side and we would have 2 fluoro, 1 iodo instead. So the answer is that there are 5 different structural isomers. And here are our 5. Let's look at another example. Pause the video and write down all of the structural isomers you can think of for hexane. Well, obviously to begin with, we're just going to have this long carbon chain of 6 carbons that's fully saturated, and that's just called hexane. And then we're going to start thinking about chain isomers, starting with the basis of pentane. And to that pentane, we can add a methyl group, but of course we can't add it on the end because then we're just back to hexane. So our first chain isomer of hexane is going to be 2-methylpentane. And then of course we could also have 3-methylpentane, which has the methyl group on the third carbon. But unfortunately we can't do 4-methylpentane because this is just 2-methylpentane flipped over. And as we know, we're always going to count that our numbers are as small as possible. So here we would just be counting from the right hand side and calling this 2-methylpentane again. So next we're going to move to a base molecule which has a chain where it's butane with 4 carbons, and now we're going to need 2 methyl groups. So again, we can't put these on the end, so we actually only have 2 options. We can either put both of them on the same carbon, in which case this is going to be 2,2-dimethylbutane, or we could put them on different carbons, in which case it's 2,3-dimethylbutane. In addition to structural isomers, we also have stereoisomers, and these are molecules that have the same structural formula, but they have a different arrangement of atoms in space. And there are some different types. There are EZ isomers, Cis-trans isomers, and Enantiomers, those mirror images we talked about before, like thalidomide. We'll start talking about EZ isomerism, or it's sometimes referred to as geometric isomerism. Now, the basis of these isomers is this carbon-carbon double bond, which is also referred to as a stereocenter. And the important thing about that carbon-carbon double bond is that it can't rotate. So whichever groups or atoms are attached to that stereocenter, they're going to stay in the same position relative to each other. So we're going to have this flat planar molecule with 4 different groups coming off the stereocenter, or they could be 4 of the same group. So there's going to be 4 of them, but you could have a molecule like this ethene where actually they're all the same. And in that instance, we're not going to see any isomerism because all of the groups are the same as each other. But as we start to have more complicated groups, or even just atoms of different types, like having halogens substituted in, then we're going to see this isomerism. So typically you might see a molecule more like this, where we've got some bromine and some chlorine and also this methyl group attached. And suddenly it makes a difference which of these groups are on the same side of the molecule as each other. We name these stereoisomers based on something called CIP priority rules. So the first thing that we need to do is identify the stereocenter. So this is that carbon-carbon double bond that has these 4 groups around it. Then we're going to think about the 4 atoms that are directly attached to that stereocenter. So even though it may be, say, a methyl group or an ethyl group attached, at that point I would be looking at the carbon atom. You're looking at what's actually making the bond. And then you're going to assign priority based on the atomic number, which always catches a few people out. It's not the atomic mass, it's the atomic number. So if we go back to our bromochloropropene, and we look at each of the 4 atoms that are bonded to that stereocenter, we can write down what each of their atomic numbers is. And then if we look at each side of the stereocenter in turn, we can say what is the highest priority atom, which is the one that has the highest atomic number. So that's going to be the bromine on the left and the carbon on the right. And you can see here that these are on opposite sides of the molecule. So that makes this an E isomer, which stands for entgegen, which is a German word meaning that they're far apart from each other. Of course, sometimes it's not going to be individual atoms and the molecule might look a little bit more complicated, but we're still going through that same process. So here with this pent-2E molecule, I can identify that the stereocenter is this carbon-carbon double bond and write down the atomic numbers of each of the atoms that is bonded to it. Now to make it a little bit easier, I've just popped it onto its side, and this makes it really, really clear that we've got one high priority group above the bond and one below it. So again, they're not together, and therefore this is an entgegen isomer, an E isomer. Now, it's never that simple, is it? And particularly when you're looking at quite complicated hydrocarbon molecules, it's entirely feasible that all four of those atoms that are bonded to that stereocenter may be carbon atoms. So in that case, you then need to look at what is bonded to them, and we kind of go one layer further out. So if we look at this molecule here, again, we can see that our carbon-carbon double bond is the stereocenter. And then on the right-hand side, we've got carbon is 6 and hydrogen is 1. But on the left-hand side, we've got carbon is 6 and carbon is 6. So we then look at what's bonded to that upper carbon atom, and we can see that we've got 3 hydrogen atoms, all of which have an atomic number of 1, giving us a total of 3. Whereas down the bottom, we've got 2 hydrogen atoms, which are 1 each, but we've also got another carbon atom, which is 6. So that gives me a total of 8. So therefore, my lower carbon is going to be the higher priority one, and my upper carbon is going to be the lower priority one. So once again, this is an entgegen isomer. Now, do watch out, because it can be easy to start thinking in terms of, oh, what's the same as each other? So here we've got a methyl group attached on the top of both sides of this molecule. But actually, if we think about those atomic numbers, well, we can see that on the left-hand side, that carbon is the high priority group, but on the right-hand side, it's the low priority group. So again, this is an E isomer. Rather than classifying an isomer, you could be asked to draw it. So here we're drawing the E and Z isomers of this molecule. So we're going to start off with this carbon-carbon double bond in each case. And then in one of our isomers, we're going to have the high priority groups on opposite sides of the double bond. And in one of the isomers, they're going to be on the same side of the double bond. And then we can fill in our hydrogens. So we've got our E isomer on the left and our Z isomer on the right. Another type of isomerism is Cis-trans isomerism, and you're going to meet this both in organic chemistry, but also when you start thinking about complex ions in transition metals in the inorganic chemistry section. So in order to have Cis-trans isomerism, you need to have different groups and 2 of them need to match. So if they're on the same side of the double bond, then this is called a cis isomer. And if they're on opposite sides of the double bond, then this is called trans isomerism, like a transatlantic flight or like being transgender. Now, it's worth pointing out that although you can have molecules with a single bond that look like cis-trans isomers, these actually aren't cis-trans isomers. And the reason for that is that that single carbon-carbon bond can rotate, and therefore the entire right-hand side of that molecule could flip over so that the chlorine is now at the top of the molecule instead of the bottom. So they actually aren't isomers of each other. If you take a second to look at your hands, these are a really good way of understanding enantiomers. So enantiomers are mirror image isomers. So they're not superimposable. If you try putting your right hand on top of your left hand, they don't match up perfectly. But if you flip one of them over so that you can put them palm to palm, then they do match. So at a molecular level, we do have right-handed and left-handed molecules, and this is really important, particularly in biology when you think about things like DNA and protein that do have a handedness. Optical isomers are said to be chiral molecules, and the asymmetric carbon at the centre of them that has 4 different groups bonded to them is referred to as the chiral centre. If you take any of the amino acids, then the vast majority of their shape is exactly the same, and it's just this R group down the bottom that changes. And any of the amino acids, apart from glycine, where this is a hydrogen, do have a chiral centre, and it's this carbon here. Now you might be looking at this and thinking, well, hold on, that carbon is bonded to two different carbons. And it's true, it is, but we need to think about the whole group. So in this example, the right-hand side, we've got that whole carboxylic acid group, whereas on the bottom, we've got that methyl group. So even though they both have a carbon bonded to that chiral carbon, the group as a whole is different. Now, these optical isomers can actually do something quite cool. So if you take just a normal beam of light, it has light that is kind of moving in all different directions. But if you pass it through a Polaroid, then you can have polarized light where everything is oscillating in the same direction. And if you take that polarized light and then you pass it through a solution that has each of these optical isomers in it, then one solution will rotate the plane of polarization in a clockwise direction, and we call this the plus isomer, and the other will rotate it in the anti-clockwise direction, and we call this the minus isomer, although we also refer to these as R and S or D and L. Thanks very much for watching, and I hope you're now feeling a little bit clearer about all of the different types of isomer that you're going to meet in the organic chemistry section of A-level chemistry. If you did find this video useful, then don't forget to like and subscribe for more A-level chemistry resources coming soon.